This is the second of a two-part lecture addressing transcription in eukaryotes. In this portion of the lecture, we're going to talk about the processing of messenger RNA which occurs in eukaryotes. Before we do that, let's consider the differences between bacteria and archaea, that should really say prokaryotes, and eukaryotes with regards to the timing and the location of transcription and translation. Eukaryotic cells have a nucleus. Transcription occurs in the nucleus, and translation happens out of the nucleus. It's different in prokaryotic cells. Prokaryotic cells don't have a nucleus, so transcription and translation essentially happen in the same place and at the same time. In eukaryotic cells, transcription happens in the nucleus, and then the messenger RNA or the other types of RNA transcripts are exported out of the nucleus. Once they're out of the nucleus, if it's messenger RNA, that's when they associate with ribosomes, and that's when translation occurs. So there are some fundamental differences in the location and the timing of transcription and translation in these two types of cells. And we see differences, a lot of differences in how messenger RNA is handled in these two types of cells. So eukaryotic messenger RNA transcripts are extensively processed. And what that accomplishes is a variety of things. One of them is that eukaryotic transcripts, and in the word transcript I mean messenger RNA, they can last for hours, whereas prokaryotic transcripts are very quickly degraded. That makes sense given the location and the timing of transcription and translation in these cell types. Also remember that prokaryotic cells are in contact with the external environment at all times. They're single-celled organisms. These organisms grow faster, and therefore they need to be able to produce proteins very quickly. So transcription and translation happen faster in prokaryotes than occurs in eukaryotes. Eukaryotes have somewhat different biology. They have bigger cells. They have more issues with, waiting on diffusion for necessary reagents for chemical reactions. Some eukaryotic organisms are multicellular, and they maintain very carefully regulated internal environments through homeostasis. All of this adds up to a scenario biologically wherein eukaryotes are able to take longer to produce protein from the gene level. So now let's think a little bit about the processing of eukaryotic messenger RNA. We've already talked about how the messenger RNA is produced, and the result of all of that would actually be called an immature or pre-messenger RNA molecule, or a nascent transcript. Right. One of the three ways in which messenger RNA is processed in eukaryotes, and the first processing step that occurs, is 5' capping. So as soon as messenger RNA comes spooling out of the transcription bubble, it's got a 5' end. Okay? And the nucleotide right at the 5' end is shown here. Okay? Note that it has 3 phosphate groups associated with it, unlike the nucleotides that have been incorporated in the actual strand. What will happen first is the most external of those phosphates will be removed. Now at the same time, there is a guanine that is being modified, and that guanine is shown here. It's not attached to anything else. What's going to happen chemically to it is 2 of its phosphates will be removed. Then what's going to happen is these are going to connect. So we're going to end up with 3 phosphates shown here. Okay. Also at the same time, or additionally, 2 other things that I want you to know. One is that this chemical bond is a little unusual. Okay. It's a 5-5 bond, and the other is the addition of methyl groups to these bases at the end, and a methyl group down here on the modified guanine. So it's a methylated guanine that is added to it. This is more chemical detail than I really expect you to remember. What I really want you to know is where the 5' cap goes. It's on the 5' end of the messenger RNA. I want you to know that that's the first step in the modification or processing of messenger RNA in eukaryotes, and I want you to know the 2 purposes for doing this. The purposes are, 1, to stabilize messenger RNA so that it lasts long enough to be translated, and 2, to assist messenger RNA in binding to the ribosome in order to initiate translation. All right, we talked about the 5' capping. Now let's talk about what happens at the other end, which is the second processing step for messenger RNA in eukaryotes. What happens at the other end, the 3' end of the pre-messenger RNA molecule, is something called 3' polyadenylation. There are a suite of proteins that play a role in this. There is no DNA template for the polyadenylation process. So the proteins are shown here and their various acronyms are shown here. They are going to recognize a particular site in the messenger RNA molecule at the 3' end. They're going to do a little cleavage or cutting, and then there's an enzyme that performs polyadenylation. And the end result is that you are going to get this 3' poly A tail formed. The 3' poly A tail helps to protect the messenger RNA transcript from degradation by exonucleases. It also helps to facilitate efficient export of messenger RNA from the nucleus out into the extranuclear environment where translation will occur, and it plays a role assisting with the termination of the transcription process itself. All right, so you guys should have already been aware of the fact that eukaryotes have introns in their genes. So introns are areas of the gene that are not specifically coding. And introns, once they are transcribed into RNA, are then subsequently removed as part of the processing or treatment of messenger RNA in order to get it ready for translation. So what you see here is a DNA gene, then a pre-messenger RNA molecule, and then the actual messenger RNA that will be translated by the ribosomes into a polypeptide. Okay? And you'll notice a shrinking in size here. So the actual DNA gene includes the promoter region, it includes some termination sequences, it includes sequence that relates to the 5' and the 3' untranslated regions, and it includes intron and exon DNA. The pre-messenger RNA molecule is smaller in that we do not see any sequence related to the promoter or any of the termination sequences that were in the DNA gene. We still have introns and exons, and we still have the 5' and the 3' UTR, and they're going to persist all the way to translation, although they themselves won't be translated. The removal of intronic or non-coding RNA is accomplished through a process called RNA splicing. And you'll notice that the pre-messenger RNA molecule is much longer than the messenger RNA molecule. So once splicing has occurred, it shrinks or makes the messenger RNA transcript much smaller. That's a mature transcript at this stage here, ready to be translated. The timing of this is such that it occurs last of the 3 stages of messenger RNA processing. So the first thing that will happen is the 5' capping, then you'll have the 3' polyadenylation, and then you will have splicing. So some ways to think about splicing. You want to remember that splicing removes introns and also connects exons. It's splicing things together. Okay, connecting or splicing exon messenger RNA together. So you could think about this, with regards to a sentence as I've shown here, and you can see prior to splicing, the messenger RNA molecule might read something like this, which doesn't really make a lot of sense, right? Looks like it's written in code. After splicing, we actually have 3-letter words that make sense. You may now tip the pot and sip the tea. It's much smaller and it's got sense associated with it. So that's representative of the actual mature messenger RNA molecule. What does splicing? Little complex that get nicknamed SNRPs. And SNRPs are made out of small nuclear ribonucleoprotein particles. In other words, they're small nuclear RNA and proteins. Rather than going through splicing in this lecture, I would like you to use the very excellent assignments and tutorials on splicing in the Mastering Genetics assignment that I set up, and refer to your text. Please take careful notes and have an explanation of this process in your notes for studying for the final exam. If you're still confused, this is an additional resource that you can refer to. Right, so now that we are starting to get a better understanding of the human genome in this current era of scientific times, we've learned a lot of really surprising and interesting things. And we've talked about these or alluded to them in some of our earlier coursework. One of them is that only a tiny amount of the human genome is actually encoding proteins. So it's somewhere on the order of magnitude of 1%. And when you look at that 1%, what we see when we study the genome using bioinformatic tools, we see that there's somewhere in the order of magnitude of 20 to 25,000 protein encoding genes. These are usually or sometimes referred to as open reading frames. They look like they're genes, okay, but according to the hallmarks of what makes a gene. Now, what's really cool is that when we try to count up the number of proteins that human cells have been found to contain, that's a much harder number to get a feeling for, and that's what someone who does proteomics work does. That we know we're still really estimating, but there are at least 250,000, probably as or possibly as many as 1 million proteins. So how do we get a 250,000 genes, 250,000, from maybe 25,000, or sorry, a 250,000 proteins from maybe 25,000 genes? So we've got one gene. So it's a 1 to 10 ratio there. Number of genes we know about, number of proteins that we think that we know about. And that's a real mystery. Now, one of the answers to that mystery is the idea of alternative splicing. And this is something that we know is true. And what is shown here is the classic example of alternative splicing, which is also in your textbook. What's shown here is the pre-messenger RNA for a gene that encodes 2 different proteins. One of the proteins is called CGRP hormone. It's a protein hormone, and it's made in neural cells. It acts as a vasodilator, and it affects pain transmission. It plays a role in migraines, so it's really well studied. The other protein is called calcitonin. It's a protein hormone as well, and it regulates calcium load. It's really important in the thyroid. Both of those proteins are encoded by the same gene. The way that you can have one gene and have more than one proteins as a result, the amino acid sequences are different in these proteins. These are distinct, different proteins, different structure, different function. Okay, the way that you can have that happen is alternative splicing. So there's one gene, there's a set of introns. Sometimes in some cells, some introns are taken out, and in other cells, different introns are taken out. And the cell is able to regulate or manage that. And this is one of the ways that we're able to use our relatively small amount of coding sequence to get a rather vast number of proteins produced at the level of the whole organism. It's really cool. So again, thinking about alternative splicing, this is how eukaryotes get more proteins than the numbers of genes they possess. This is a way to maximize the genome. It promotes biodiversity, so it's evolutionarily favored. Now, the dark side in all of this is that in humans there are many mutations that give rise to disease that are mutations that affect splice sites. So sometimes when we don't splice according to the plan, we can get altered proteins that produce or are associated with disease phenotypes. All right, the timing of all of this. So what we see for many genes is the order shown here. RNA synthesis begins and it's occurring. As it's occurring, you have 5' capping. If it's a large gene, you can see splicing happening as it's being transcribed, which is what's shown in this order here. Then you'll have termination followed immediately by polyadenylation and then export from the nucleus. However, in many genes, it's worth noting that the splicing can actually happen here. Okay? So sometimes, particularly in smaller transcripts, splicing will happen after everything else is complete and immediately before export. So in those cases, it would be 1, 2, 4, 5, 3, and 6. And that concludes this lecture.